During crossing over, segments of these chromosomes can exchange genetic material, leading to new combinations.
In practice, the F2 3-to-1 ratio and the 1-to-1 test-cross ratio are never achieved. Usually, 4 phenotypes are invariably produced. This is because the total linkage is rare. Most breeding experiments involving linkage produce approximately equal numbers of parental phenotypes and a significantly smaller number of phenotypes showing new combinations of characteristics, also in equal numbers. These latter phenotypes are described as recombinants. Why do you think recombinants are always few? Are the chromosomes shy or just avoiding mixing business with pleasure? In the previous linkage video, we had a 1-to-1 ratio for the test cross. However, today, we are going to see that total linkage is not possible and recombinants are produced in small equal numbers. If linked genes always stayed together like best friends, which pair of school friends in your class would represent them? And who would be the recombinant troublemaker? We carried out this example when we were dealing with linkage, and we obtained all broad abdomens and long wings. Broad abdomen and long wing, dominant over narrow abdomen and vestigial wing. We are now going to test cross and be involved in crossing over to see what we shall obtain. Now carry out a test cross using crossing over. Crossing over always happens during gamete formation. We did not show it in other crossings because it had no effect. Parental phenotypes, we have that. Parental genotypes. This is a test cross. We use a heterozygote versus a double recessive. Meiosis leads to the formation of gametes. We start with the replication of chromosomes. They double like this, on both sides. This is followed by crossing over between non-sister chromatids. If your parents gave you only their exact traits with zero mixing, would you still look the same, or would you demand a crossover refund? Let us remind ourselves of what happens during crossing over. Here, these chromosomes exchange portions of non-sister chromatids. It occurs on both sides. The result is like this, with gametes that show recombinants. On the other side, the gametes remain the same since the genotype is double recessive. Simplify it, we write all will narrow abdomen and vestigial wing. Get more information from self-study A-Level Biology. It has all the topics combined, with examples and scenario-based questions. We have the gametes, random fertilization, test cross genotypes. We come up with such a combination. Test cross, physical appearance. Broad abdomen with long wings. Here, a narrow abdomen with vestigial wings. These are in large numbers in equal proportions. They resemble parents, therefore known as parental types. We have other combinations, a narrow abdomen with long wings, and also a broad abdomen with vestigial wings. These are formed in small, relatively equal numbers. They are the new combinations, known as recombinants. They are formed as a result of crossing over. If crossing over was a dance move, which chromosome do you think would mess it up first? The one that forgets the steps or the one that insists on freestyle? Let us first remind ourselves of what goes on during crossing over. In meiosis 1, homologous chromosomes align close to each other to form a bivalent process known as synapsis. These are called sister chromatids on the same centromere. These are non-sister chromatids. They then become intimately wrapped around one another. They become intertwined, and the non-sister chromatids of the homologous chromosomes exchange portions at a point called a chiasma. This is called crossing over. During synapsis, homologous chromosomes wrap around each other so tightly, should we say they're studying biology or practicing wrestling. The chromatids finish up in separate gametes and, after fertilization, give rise to new combinations of alleles in the offspring, recombinants. The number of chiasmata formed in a bivalent during meiosis, due to the length of the chromosomes, is proportional to the number of possible combinations. This causes a genetic variety. Crossover value. This is the frequency at which crossover events occur between two specific loci, positions, on a chromosome. The number of chiasmata formed in a bivalent during meiosis, due to the length of the chromosomes, is proportional to the number of possible combinations. This causes genetic variety. It is often expressed as the percentage of crossover events observed between two genetic markers during meiosis. Using the example we discussed, the crossover value is given by the number of recombinants divided by the total number of offspring multiplied by 100. This gives us 3.5%. Crossover frequency reflects the relative positions of genes on chromosomes. Because the further apart linked genes are on the chromosomes, the greater the possibility of crossing over occurring between them is the greater the crossover frequency. The distance between the gene for the size of the abdomen and the length of the wing is 3.5 units.